Hello everyone, and welcome to our online lecture series for Bio1. Today, we finally moved on past our discussion of the nervous system and are instead going to be focusing on another system that relies heavily on signaling, this time the endocrine system. The endocrine system is made up of a series of glands or cells that secrete substances. These substances are signaling molecules. And in order to understand the nature of these signaling molecules, or hormones, we're going to need to revisit some basic principles of chemistry. Now, earlier in this class, when we talked about our macromolecules, we talked about things that were hydrophobic and hydrophilic. Now, previously we've talked about the fact that these hydrophobic things are nonpolar, they have no charge, they're neutral all around, they are oftentimes lipids, and do not mix with water. Hydrophilic things, in contrast, have some sort of charge, are polar, do mix with water. Now, there's going to be two major consequences of hormones that are either hydrophobic or hydrophilic. Hydrophilic ones are going to be able to move alone in the bloodstream because of the fact that they are dissolved in the blood itself. They are able to be broken down quicker. And this has to do with the things that break down substances in our bloodstream. As you may know, there are tons of proteins floating through your bloodstream and in all of the cells of our body. And a lot of these proteins have one simple job: recycle. Break things down that you're not currently using so that way you can use them for a purpose that you need right now. This kind of running on a treadmill is very common in a lot of organisms, as it's very difficult to store things for long periods of time. Hydrophilic substances are broken down much quicker because they are dissolved within the liquid of the blood, and as most things are hydrophilic in the body, I mean, we all are like 95% plus water, the amount of machinery devoted to breaking down hydrophilic things is much more prevalent. Contrast this with what we see in hydrophobic things. They oftentimes require some sort of chaperone, or they move in their own bubble, as they are sometimes lipids. And then their time of survival is much, much longer. Their breakdown is slow. Because when you think about it, if all of the breakdown machinery is dissolved in water and you have something that isn't dissolved in water, it might take a while for that breakdown machinery to bump into something that can help it break down. These two major differences, kind of grouping them together as one because of the fact that they both have to do with whether they're moving alone and breaking down. And then the second difference that we need to consider when talking about our hormones is where and how they act. Hydrophobic things, as we've talked about previously, can pass through the membrane with no problem. The membrane itself is a phospholipid bilayer, and if you are of similar chemistry, you should be able to ghost right through the walls. In contrast, hydrophilic things need to stay outside of the cell. They signal inside. therefore starting the cascade of signal transduction, as we talked about earlier in this class as well. So we need to consider how the chemistry of hormones changes both their function and their duration. When we talk about our hormones, we can look at this ladder of interaction. Your gland cell will produce the hormone. The hormone itself will always have some sort of target cell. On that target cell, you have a receptor specific to that hormone, or else it is not perceived. For example, you could douse your, let's say, brain tissue with a hormone specific to your kidneys, and it would not have an impact. You need to have the correct receptor on those cells. Once the hormone binds to the receptor, it will cause some sort of signal transduction within the cell. And now, based on what type of hormone, what cell was receiving, you'll get much larger downstream effects. These can be temporary, like the raising of blood pressure for a short bit, or permanent, meaning that they're part of the developmental pathway. Like, we're going to start making beards now. So all of our hormones, whether they be hydrophilic or hydrophobic, follow this general blueprint, but their downstream effects will change based on the chemistry and the target cells. All right, so let's talk about our 3 classes of hormones now. Our first group of hormones are lipid-derived. Lipid-derived hormones have the unique ability to diffuse across the membrane. Meaning that they don't need a receptor to be picked up, they just go. They don't ask permission to enter, they just enter. This obviously can have some very dangerous effects downstream, can have some very unanticipated effects downstream, so we need to be careful when applying or using lipid-derived hormones, because they will go anywhere and everywhere. They're oftentimes derived from cholesterol. And that means that in the synthesis of these molecules, in the production of these molecules, the early precursor is often cholesterol, which we classify as a steroid hormone. Now, cholesterol can be turned into many, many things. For example, cholesterol in males can be converted to the hormone testosterone. You'll notice a very similar shape here with the rings and the pentagon, or the hormone estradiol, again, rings and a pentagon. So we have cholesterol either turning into the male determining hormone of testosterone or one of the critical female hormones, estradiol. The major features of being cholesterol derived is that these molecules are insoluble in water. As we mentioned above, this prevents them from bumping into the breakdown machinery, the ubiquitination machinery. That means that they stay in blood longer. For example, if you were to look at a metric called the half-life of a steroid hormone compared to, say, adrenaline, which is a non-steroid hormone. These lipid-derived hormones can survive anywhere between 60 to 90 minutes in the bloodstream, whereas adrenaline will survive no more than a minute. This means that these steroid-derived hormones are going to be doing their signaling everywhere for much, much longer, in contrast to the very short window that adrenaline has to act. And that's probably pretty good as a biological system, because you do want some hormones that have a longer effect, and you want some hormones that may be very quick. Not always do you need to be in an adrenaline rush. That is metabolically very expensive. So adrenaline needs to come in and then disappear quite rapidly. And this class of lipid-derived hormones are the ones that we think of primarily as our hydrophobic hormones. Our other two classes are generally regarded as hydrophilic. The first are our amino acid-derived hormones. Now, amino acid-derived hormones oftentimes have the suffix INE, or in some capacity, and they have a precursor of some sort of amino acid. So a single monomer of a protein will go to build these amino acid-derived hormones. That means they generally are fairly small. And so that is another characteristic that's shared between either the steroid hormones or the amino acid hormones, is that they are both relatively small compared to our other type of hormone. Some good examples of these amino acid-derived hormones include epinephrine, the precursor to adrenaline, so the thing that gives you your adrenaline rush. It comes from the amino acid tyrosine. And therefore, if you want to understand the chemistry of epinephrine, you have to understand the chemistry from which, the molecule from which it is derived, tyrosine. And so tyrosine is one of our 20 amino acids that are used to build proteins. It's considered a non-essential amino acid because our body is able to make it. But tyrosine itself has a polar side group. You see that with the OH there on the end. Now, tyrosine has a very interesting story. It is actually derived from tyros, meaning cheese in Greek, because it was first discovered by a German chemist in casein from cheese. So, obviously, I have strong feelings about the amino acid whose name derives from cheese. Like me some cheese. And while it is a amino acid that has a polar side group, it is not. It is classified amongst the hydrophobic amino acids, though it is more hydrophilic than phenylalanine. So there's obviously a ranking here as to how hydrophobic or hydrophilic they are. But ultimately, what this means is that the fact that tyrosine itself has a polar side group and epinephrine is derived from it, it is more likely to be one of these molecules that is hydrophilic in its overall tendencies, particularly when compared to the lipid-derived hormones. And epinephrine is, of course, just one of many different molecules that are made from tyrosine. You also have the very important neurotransmitter dopamine being also made from tyrosine. And dopamine functions in many different ways in the brain, but overall it's a reward anticipator. So there's a little bit of tie in there between the molecules that give you the adrenaline rush and the molecules that make you seek reward and perhaps are associated with different addictions. Another example of a different amino acid derived hormone is melatonin, the hormone that we use that is synthesized when we want to go to sleep. So this is your sleepy time hormone. In fact, I do occasionally take melatonin as a means of being able to fall asleep. Melatonin derives from tryptophan. And tryptophan, again, is one of our amino acids. And tryptophan, again, is, or rather not again, but instead of being a non-essential amino acid, tryptophan is an essential amino acid, meaning that we do not make it, but we instead need to eat it. And tryptophan is a precursor to some very, very important hormones, both in plants and animals. Tryptophan makes serotonin. So it is. Tryptophan is a precursor to serotonin. It's also a precursor to melatonin, and it's a precursor to the auxin class hormones, which, if you happen to be a plant biologist, auxins are some of the most abundant and most important growth hormones in plants. Now, altogether, these amino acid-derived hormones will oftentimes pick up some of the chemical characteristics of the amino acids from which they are derived. On a scale of hydrophilic to hydrophobic, they will be much more hydrophilic than the lipid or steroid-derived hormones. But each of the amino acids themselves can have some hydrophobic tendencies. And that will obviously change what the interaction domain or the permeability or the transmissibility of these different hormones that these different hormones have. So it's important to keep in mind what the precursor molecules are always. Now, in looking at our last class of hormones, we have a slightly different precursor group. Now, these are differentiated by their size. These are our peptide-derived hormones. And you guessed it, rather than being single monomers, peptide-derived hormones are instead short polypeptide chains. Now, because they are going to be short polypeptide chains, they oftentimes are secreted proteins that derive from that very, very useful packaging plant within the cell, the Golgi body, that does all the cleaving and synthesis and prepping that is necessary to make these different larger hormones. They, partly because of their size and because they are water soluble rather than hydrophobic, they're not soluble within lipids. Peptide hormones cannot pass through the plasma membrane, therefore these also need receptors on the surface of the cell. As three examples of these different peptide-derived hormones, we first have oxytocin, a very important hormone in the process of labor. We have the human growth hormone, and then the follicle-stimulating hormone, or FSH. This is the one that will go ahead and cause your ovaries to release an egg or stimulate your follicle so that way you can begin the menstrual cycle. Again, these are much larger, but they still have the generally hydrophilic and insoluble in lipid properties that we see in amino acid-derived hormones, and therefore will have a much shorter half-life within the cell altogether. Now, when we talk about these different hormones and their different modes of actions, there's a few things that we need to consider in terms of how sensitive cells are. Now, because hormones always bind to a receptor, they're going to bump into many of them, but they're only going to work with one specific type of receptor in the target cell. Therefore, the number of receptors on that cell will always be proportional to sensitivity, or how much of the hormone is needed in order to elicit a response. This leads to a very, very simple downstream regulatory effect. If you want your cells to be more receptive or upregulate, all you got to do is make more receptors. And if you want to downregulate the impact of that hormone, whoops, nowhere did I say that I wanted to spell things correctly. Downregulation, except for I definitely do, so I apologize for that. If you want to see downregulation of the hormone, all you need to do is have less receptor. And so let's talk about the differences between these three different classes and the types of receptors that they will interact with. When we look at our lipid hormones, we're going to be working with intracellular hormone receptors. And that means that these receptors are actually present within the cell. Oftentimes, these intracellular hormone receptors take these lipids with the carrier or chaperone protein. But they're going to pass through the plasma membrane, infiltrate the cell, and directly cause changes in gene expression, or gene expression. So your steroid hormones don't need to ask permission. They go straight to the DNA and cause their changes. In contrast, both our amino acid and peptide-derived hormones need to have plasma membrane hormone receptors. Because they need to be doing that cascade that we see. They're always going to bind at the plasma membrane and then cause some sort of signal transduction within the cell. And oftentimes, these molecules that are doing that signal transduction or communicating within the cell are our G proteins, those molecular switches. Because these use plasma membrane hormone receptors, they always have a primary messenger. The primary messenger being the one that's outside the cell and the hormone itself. And a secondary messenger is oftentimes the messenger that moves on the inside. So to better understand this, let's go ahead and take a look at one of our amino acid derived hormones, keeping in mind that amino acid and peptide derived will both be using that plasma membrane. And let's see exactly what the downstream impacts inside the cell are. So here we'll be talking about the hormone epinephrine, or precursor of adrenaline or noradrenaline. Epinephrine needs, or as a amino acid derived hormone, it will need a receptor at the plasma membrane. In this case, one of the possible receptors is called the beta androgenic receptor. The beta androgenic receptor is bound to a G protein on the inside, which again uses GTP and GDP as its signaling molecules. When those GTPs are converted to GTPs, This impacts an enzyme called adenylyl cyclicase. And adenylyl cyclicase is going to induce lots of phosphorylation of different kinases. So adenylyl cyclicase will go around and turn on something that then goes on and turns on a lot of other effects. The secondary signaling molecule here is cyclic AMP, because cyclic AMP will cause the cell-specific response based on the cell that has this beta-androgenic receptor. And of course, as we've talked about, just like in eyesight, one of those hormones that causes or deals with signal transduction, this is oftentimes a package. They oftentimes come together. It's a modular approach. We again see our phosphodiesterase. Let's come back again. But this is only one model whereby you have an external hormone bound to a receptor causing a change in signal of a G protein that has a secondary signaling molecule that then induces a cell-specific response. But what happens if you're in a different cell? What happens if you have a different cell receptor? Well, adrenaline, or in the same family as epinephrine, has different receptors based on the different type of cell you have. So you're not always going to have the exact same response in different tissues in the body. For example, in some tissues you will have a beta receptor, just like we see here. In some cases you will have an alpha 2 or an alpha 1. But note how every single time they are connected to a G protein, that molecular switch inside of the cell. Now, downstream of that G protein, you have different target enzymes. For example, adenylyl cyclicase or phospholipase. And phospholipase, with its secondary signaling molecule of calcium, will actually cause smooth muscle contraction in smooth muscles. So this is what the reaction that we're seeing in the bloodstream. So in the bloodstream, you're seeing smooth muscle contraction to actually get you to have a higher blood pressure, right? In those cells with beta-androgenic receptors, you might see heart muscle contraction. So your heart has receptors for adrenaline, which makes sense. Adrenaline causes your heart to beat faster. In different tissues, because they have the beta receptor, you're also going to see smooth muscle relaxation, or even glycogenolysis. I said that slowly because glycogeno, as in glycogen, lysis, or the breakdown of it. In some tissues, you will be seeing the production of glucose from glycogen in order to make energy more available to you. And in this way, one hormone or one group of hormones, when paired with different receptors, can have totally different effects on different parts of the body, despite the fact that they oftentimes use similar features. So this is a very modular approach, meaning that we're always seeing, or very commonly seeing, the same players, some sort of hormone on the outside, some sort of receptor on the surface that's coupled with a G protein, and then some sort of secondary messenger, second-degree messenger, that then causes your downstream gene expression changes. But you have to know exactly what tissue you're looking at and exactly to understand the exact effect of what is going on. Oh, I did forget to mention that. How does this whole thing turn off? Well, that's the role of that phosphodiesterase. Phosphodiesterase goes ahead and deactivates cyclic AMP to AMP, thereby telling the cell that we need to start restocking our AMP to ATP and refurbish from that adrenaline rush. And so this is going to be kind of the blueprint that we go over for the rest of the day. We've got a couple more hormones to discuss, a couple more glands to discuss. So that way you begin to understand how the downstream cellular signaling patterns will relate to the overall organismal response. And for our next example, we'll dive into the regulation of water within the body, or osmotic regulation. With the pair of hormones, antidiuretic hormone, ADH, and aldosterone. So this image from your textbook puts it all into quite stark contrast. It's a process by which renin, angiotensin, and aldosterone increase blood volume and blood pressure. So let's follow the arrows. Angiotensinogen is the hormone that generates angiotensin. It is made in the liver. Angiotensin uses the intermediate of renin. Renin being produced in the kidney. This will get converted to angiotensin 1 and angiotensin 2. Angiotensin 2 has some direct effects. Angiotensin 2 causes arteries to constrict, meaning that you have a rise in blood pressure. It decreases the amount of filtration occurring in the kidneys. We'll go over this later. Therefore causing you to retain more of your water. And then it also does this clever thing where it increases thirst. So you're going to get more thirsty. And then there are two different hormones that will play a downstream effect from angiotensin 2 as to what exactly they are doing for osmotic regulation. Those two, again, are aldosterone and ADH. ADH is made in the hypothalamus, and aldosterone is made in the adrenal glands, which are little hats on top of your kidneys. Aldosterone is a hormone that's responsible for the reabsorption of sodium and potassium. Being made near the kidneys, it makes sense that it's going to hit the solute side of things. Meaning that it's impacting how many electrolytes are in your bloodstream. With more electrolytes, you need to pump more water into the bloodstream in order to have that same dilution levels. ADH is a little bit different. Rather than hitting the solute side of things, ADH is hitting or impacting the water excretion from the kidney. It mediates how many aquaporins are in the nephron collecting ducts. So you're literally adding water reabsorption pipes. So that way less water makes its way into your urine. Your urine will be more concentrated. It will always lead to more water reabsorption. Antidiuretic hormone. It is a not diuretic. It causes you not to pee as much because you're filtering out more water, and therefore your urine is more concentrated. Now, both of these rely on the osmoreceptor in the hypothalamus. Or rather, ADH relies on the osmoreceptor in the hypothalamus because it's measuring electrolyte content. Why do electrolytes and water, or what do electrolytes and water have in common? Well, if you always want a 95% water, 5% electrolyte mix, and you see that the electrolytes bump up to, say, 7%, don't you always know that the water mix is at 93 and therefore you need more water to bring in order to balance things out? Yeah, so by measuring the electrolyte side of things, ADH can actually control the amount of water in your blood, which is kind of clever. ADH, again, is doing this via aquaporins, whereas aldosterone is impacting the absorption of ions. But both have a role within your water filtering organ, the kidney. Now, when you have too little ADH, or you're not producing enough antidiuretic hormone and therefore are not reabsorbing enough ions from your urine, you will see something called diabetes insipidus, a different form of diabetes that does impact your overall body function. It will eventually lead to dehydration. And too much urine. So how does that happen? Well, if your body doesn't make the correct amount of ADH, you aren't absorbing enough water because you don't have the correct amount of aquaporins. And if you aren't absorbing enough water, you're probably peeing out too much of it. But you're peeing out so much that it's actually causing dehydration within your body. So it's so incredibly important that this system of aldosterone and ADH are both balancing the electrolytes in your bloodstream and the amount of water retained in your system. Because either can have a negative impact on the physiology of the entire organism. So next up, I want to go over the things from the chapter that I don't expect you to go over. We won't, on our exams, be focusing on sex hormones and their signaling. No discussion of the growth hormone and its signaling. And then we won't talk about calcium. But I'd encourage you to potentially read those sections in the chapter, particularly if you are going into some sort of medical field. We don't have time to go over them, but they obviously will show up on your medical exams at some point. So don't leave yourself any gaps. For our next discussion, I want to focus on metabolism. That thing that we all wish we had faster of. Because then I'd be able to eat cookies without consequences, right? Your metabolism, or your regulation of your metabolism, corresponds quite greatly with your glucose levels. And these glucose levels change a lot throughout the day. Whenever you eat a meal, you can spike your blood glucose levels, and then you better hope that you have the hormones needed to hide that glucose so that way it doesn't cause damage to your kidneys. The hormone that does this is, of course, insulin. Insulin is a hormone produced in the beta cells of the pancreas. like to throw in this little image to illustrate how we know that it happens to be made in the beta cells of the pancreas. So this is a bit of data that derives from Wikipedia, but what scientists do here, this is called RNA expression. So we're looking at where insulin's RNA is made and therefore where its hormone is made. Keep in mind that it is a peptide hormone, so you're going to be making it in the Golgi body and secreting it. And we see that across many different tissues of the body, like whole blood or whatever the cerebellum is, that's part of your brain. It's not made in the cerebellum. Or say the lungs or the prostate or the thyroid. It's not made in any of those. But then you come to the pancreas. And the pancreas and the pancreas islet are both tissues in which insulin RNA is made. So that's how all of that work is how science determines that insulin is made in the beta cells of the pancreas. Kind of a lot, right? Insulin is made in the beta cells of the pancreas in us humans. From there, it will be transported into the bloodstream. Oops. And while it's in the bloodstream, it can increase glucose uptake in cells. Oops. In the red blood cells, the way in which it does this is it causes the localization of the glucose transporter 4, or GLUC4, move to the plasma membrane. And when GLUC4 is on the plasma membrane, glucose is being brought into our red blood cells. But not only is insulin impacting transport into red blood cells, but it's also increasing transport of glucose into muscles and the liver. In the liver, you're actually going to be able to store that glucose as glycogen and then fat. And this cascade of converting blood sugar to storage sugar to fat is why an excess of calories will eventually cause an increase in fat in our tissues. Now, the way in which this transport occurs is again to do with that sensitivity. By producing insulin, you change the number of receptors of GLUC4 on the plasma membrane. By doing that, you move in more and more glucose into the cells. Why do you want to be doing this? Well, because glucose is a small, sharp sugar molecule that if it's in too high concentrations in some very small blood vessels, like within your kidneys or next to nerve cells, your nervous tissue, that glucose damage will increase, ultimately causing glucose to penetrate your kidneys, and at first glucose will go through, but eventually blood may enter into the urine. This, of course, is the failure of insulin is actually a failure of the beta cells and will lead to diabetes. Diabetes mellitus. On the other side, just like insulin is a regulator when you have too much blood sugar, we will also make a different hormone that contrasts with the function of insulin when you have low blood sugar, called glucagon. Uh-oh, the glucose is gone. Glucagon is secreted from alpha pancreatic cells, insulin beta, glucagon alpha, and instead of hiding away glucose and producing glycogen and producing fat, as insulin does, glucagon is going to release glycogen and break down fat. But again, biochemically, glucagon is only going to be made when you have too little blood sugar. So if you want to be breaking down fat, you need to have burnt off a lot of your blood sugar. Exercise. I should probably go do that soon. I will be going for a run after I'm done recording this. Next up, on our discussion of different hormones, we have both a hormone and an essential element. Here, in terms of metabolism regulation, we have the complex of thyroxine and triiodothyronine. One of those real good Scrabble words. Thyroxine and triiodothyronine, or T4 and T3, much easier to say, combine to determine your basal metabolic rate. Or how much energy you're burning by just chilling. T4 and T3 are transported across membranes and bind at the mitochondria. Now, why is that unique? Well, a few things first. T4 and T3 are both amino acid derived hormones. They actually come from the same pathway as tyrosine. This means that they should not be, or regularly wouldn't be entering the cell, right? They have some sort of secondary molecule. But T4 and T3 are instead shuttled in by a carrier protein and directly bind to the mitochondria and nucleus. By binding to the mitochondria, you can ramp up glucose breakdown. You can start making or breaking down more and more glucose. In doing so, you can make more heat. Now, in order to make T4 and T3, you actually need a very specific element: iodine. The production of T4 and T3 is actually triggered by thyroid stimulating hormone, or TSH, that then will go to the thyroid itself and begin producing these amino acid derived hormones, but they need different amounts of iodine. T4 needs 4 iodines. T3, guess how many it needs? 3 iodines. And if you do not have enough iodine in your diet, you may experience something called a goiter. A goiter is the swelling of the thyroid gland because it's not having enough iodine to produce these hormones and instead kind of gets filled up with all the production machinery, but not enough iodine. Now, thankfully, in developed countries where salt has iodine added to it, we don't see a lot of goiters. But this does not mean that we don't have thyroid gland problems. In fact, the manufactured form of thyroxine, called levothyroxine, is the most prescribed drug in the US, with over 100 million active prescriptions. That's a lot. A lot of people have problems with their thyroid where it will not produce enough T4 later in life, and therefore their metabolism's slow, they become more lethargic, and they blame it on age. When really, it's not age directly, but rather a shortage of T4 hormone. My grandma was one of these individuals, started feeling a little bit sluggish at the ripe old age of 80, thought that her time was coming, her time was nigh, but went to the doctor, found out that her T4 levels had dipped, was on a mild prescription for low amounts of levothyroxine, and bam, zippy grandma again. So please be aware of the fact that these hormones can have a major impact on your overall energy levels. And it does help to understand how levothyroxine will go to produce the same downstream effects of glucose breakdown and more heat and more rapid metabolism, because these hormones have receptors at practically every cell in the body. And that makes sense, because every single cell has mitochondria, and since T4 is going to look to bind at your mitochondria and your nucleus, every single cell should be able to pick that up. And now, not only is T4 actually impacting the mitochondrial rate, but we also see the rate of glucose breakdown, but we also see protein metabolism start kicking up. So that's one of the effects of it binding to the nucleus. T3 has some other impacts downstream. You'll see that it can kick up brain development. It can make your body more sensitive to adrenaline. It can increase the breakdown of, or rather the catabolism of proteins and carbohydrates, as well as increase your heart rate and breathing rate. So overall, these are kind of your master regulators of your metabolic rate. Now, the last hormone that we'll go over as a group, just very, very briefly, is the hormone that causes, that is produced in long-term stress response. And so you will notice that there are actually negative effects to being stressed for extremely long periods of time. And I'm sure that I'm not helping that. Or rather, a lot of classes don't help out your stress, but I do hope that you get to a point where the stress is manageable and actually productive rather than detrimental. Because when you have long-term stress responses, you'll produce the hormone called cortisol. And cortisol has anti-inflammatory effects. But also immunosuppressive effects. So you may notice that during times of high stress, your immune system may get weak. Your adrenaline and all the other hormones that are ramping you up will cause you to be highly alert, But then after the stress has subsided, you may get sick. I historically would frequently get sick after finals week because of the amount of stress derived there. Now, cortisol also causes long-term fat storage because you think that you are going through a stressful time and any calories need to be conserved for afterwards. So, with that, I'll just end the lecture by showing y'all the table 37.1. I do ask that you read through this, but pay very close attention to the hormones that we have highlighted, as they're the ones that we talked about in this class, like the antidiuretic hormones synthesized in the hypothalamus and what it does for water reabsorption, like our again, we skipped the discussion of growth hormones and many of those that are found in the pituitary, but we did talk about thyroxine and triiodithyronine, both made in the thyroid. We talked about aldosterone and cortisol. Whoop, highlighted the wrong one there. Cortisol. And then, of course, we talked about insulin and glucagon. So please spend some time reading the definitions at the end of this chapter, as they will certainly help you solidify your understanding of these different hormones. But always keep in mind when you're talking about these hormones that their class, whether they're lipid or amino acid and peptide derived, will have an impact on how long they stay in your bloodstream and how they impact the cell. Meaning, do they send something in that signals, or are they the signaling molecules themselves?